Dr. Rooney, are you there to introduce your protege?
Yes, if you can hear me.
I can hear you.
Okay, very good. Our next speaker is Jales Fonseca. Jales is a native Brazilian who got an undergraduate degree in Sao Paulo and then got a master's degree in plant breeding. I believe it's plant breeding at Federal University of Lavras, and came to our program in summer of 2017 to get a Ph.D. in plant breeding as part of some funding from the Monsanto, at the time Monsanto, to facilitate training of students in plant breeding from South America. Over the past 3 years, he's developed his project, and it's become quite a important part of our program. And today he's going to speak to y'all about a few things that he's been doing that are part of his genomic selection project in sorghum.
All right, can you hear me?
Can hear you, yes.
All right, let me share my slides here. All right, thank you, Dr. Rooney, very much for your introduction. And today I'll be talking about genomic selection and prediction in hybrid crops, and I'll be using sorghum as a model species. And I hope that by the end of this presentation, you guys get more familiar with some of the different models that we can use to perform genomic selection, and how you can implement genomic selection within a sorghum breeding program pipeline. So sorghum is a self-pollinated species, and it has been grown as a hybrid crop since the development of the cytoplasmic male sterility system. And since there's that discovery, the sorghum breeding program pipeline has altered in order to create heterotic groups and breed for the female side and the male side in order to create better hybrids at the end. And it also facilitated the development of the hybrid sorghum seed industry. Although the yield after hybrid development increased, the rates of genetic gain has been limited. And in order to solve some of these problems, some researchers had introduced some new methods to increase the rates of genetic gain, and those are based on predicting hybrid combinations. Some of the strategies were based on crossing the best line per se, or using genetic distances in order to maximize heterosis, or using test crosses to estimate the best lines in hybrid combinations and then put those lines together to increase the prediction, the yield potential of the hybrid. Although these methods were not successful in explaining or predicting hybrid combinations, it was successful to increase the means of both heterotic groups, males and females. He ended up increasing the yield production. And then another way to predict hybrids was introduced by Bernardo, and he actually was using information from the general combining ability of the female, the general combining ability of the male, and the specific combining ability. And also, he showed us how to put some pedigree information into these models. These models were not tested in sorghum yet, and probably because of the focus of the sorghum breeding program, which is based on releasing new lines and not actually putting the best hybrid combinations together. But so we can use that type of blood prediction to use in a phenotypic selection and also in a genomic selection. So it's hard to beat phenotypic selection because you always put your genotypes in the ground and then you can select them based on their performance. But it's very unlikely that you will have enough resources to test all the possible hybrid combinations across all the possible locations. So that's why people are trying to increase the rates of genetic gain by developing genomic prediction models in order to predict the hybrids or lines that were untested. And basically what you do, you genotype your population, your reference population, and you phenotype your reference population. And phenotypic information here is extremely important, and that will determine the successful of your predictions later on in the program. The problem is developing those genomic prediction models are not straightforward because usually you have a lot of molecular markers and not enough data points in order to regress and put the marker values specific to each marker. So we need a more robust and more flexible models that can deal with the course of dimensionality problem. So here in this example, we might have 30,000 SNPs and we only have 1,000 records in order to model all these molecular markers here. So we need to go for a more flexible and more advanced models, which includes the regularized regressions and shrinkage of estimates, variable selection, which is Bayesian, or we can use both. Just a brief description of a GBLUP model here. You can have your phenotype, and then this is the intercept, and this is the marker effect. So you can use the marker effect directly in the model, or you can use a genomic relationship matrix where you use the markers to create a relationship based on the covariance between those markers. And then you can input that information into your model, and that increases the efficiency of your model and decrease the time for you to calculate all the marker effects. So you're basing on your predictions relies on how close related your material is. According to the literature, there is several different models that you can actually develop. And the most efficient, according to the literature, are the Bayesian models, because you can include prior knowledge to create hyperparameters that allows you to model specific markers using simple variances. So you can simple variances and put the effect of a marker separately, which makes this model a little bit more flexible. Okay, enough about genomic prediction only, genomic prediction models, because we also need to input all that information into a breeding program pipeline. And when we do that, we need to account for the environmental effect. We need to be aware that phenotypic data is extremely important. Again, I can't stress that enough. So I need to have good data. And then we can also create genomic prediction models to predict breeding values or to predict hybrid performances. So you need to develop models specific based on the goal of the breeding program. And also, an important effect for you to keep track on is the population structure. And there are some strategies for you to go and to apply. And in hybrid crops, for example, where you have three different sources of variation, which is the GCAs of males and females and the SCAs. Putting the population structure is a good thing for you to increase the reliability of your model. And a major challenge is how can we model a G by E using in a context of genomic prediction models. So this is why I've been trying to do, and one of the objectives of this presentation is to predict the performance of grain sorghum hybrids using elite inbred lines in two forms. We'll be using a genomic model and a non-genomic hierarchical Bayesian model. We're using the Bayesian framework. Specifically, we'll be comparing a kernel-based Bayesian GBLUP with a non-genomic Bayesian ridge regression model. And at the end of the presentation, I will propose a conceptual framework for implementing genomic selection in the sorghum breeding program pipeline here, Texas A&M. So for us to do that, we select 10 elite inbred lines in Kansas and 10 elite inbred lines here at Texas, a total of 20 parents, being 5 female and 5 male. And those are displayed here in this picture. And by using a factorial design mating scheme, we were able to produce 100 hybrids. And those 100 hybrids were divided in 25 hybrids, meaning that we have a Texas by Texas hybrids, Kansas by Kansas hybrids. Then we have a Kansas as a female and a Texas as a female in hybrid combinations. All these 100 hybrids were tested in 10 different environments across Kansas and Texas. So in 2018, we had Rio Farms, Victoria, College Station here in Texas, in this area here, and Garden City and Colby up there in Kansas. 2019, Taft, Victoria, and College Station, and again, Hayes and Colby. We were evaluating 3 important agronomic traits for sorghum, which is grain yield, plant height, and days to antithesis. And we developed genomic prediction models for all of those 3 traits. So for the phenotypic analysis, we used the linear mixed model, assuming hybrids as fixed in order to calculate the best linear unbiased estimators, which are the means, to train our genomic prediction models. We're going to call this is a first step of genomic prediction development. And we also used this model, assuming hybrids as random effect, to extract the variance components and calculate our repeatability. Similarly, we did the same thing for a multi-environmental trial across all the 10 environments. And we used the G plus E plus G by E. We only separated this because we used the same model to create our genomic prediction. So our G is composed by the GCA of females, GCA of males, and the SCA of the hybrids. And our G by E is just those effects multiplied by the environmental effect. We use the linear mixed model again to extract the variance components, calculate the reliability, repeatability, and the percent coefficients of variation. For our genotypic data, we extract DNA from all of those 20 parents, and using NGO restriction enzyme, we were able to find 117,000 SNPs that, after cleaning the data, following all the high quality control steps where we delete all the missing values and we delete the marker allele frequency, the minor allele frequency, we ended up with 35,000 high quality SNPs. And all of these SNPs were used to calculate the genetic distances between those 20 parents. And we plot those distances in a principal coordinate analysis. Also, we used those 35,000 SNPs to calculate the genomic relationship of the parents. So we calculated the genomic relationship of the females, genomic relationship of the males, and we used those two matrices to calculate the genomic relationship of hybrids using the Kronecker product. That's how we included the population structure in the genomic models. So this is the most important part of the project where we actually talk about the models. And again, we're going to be applying genomic selection, applying prediction of hybrids using genomic information and non-genomic information. And we're going to make predictions for a single environment, multiple environments, and multiple environments with G/E. And these are the effects we're considering within each model. For our G, we included again the GCAs of females, males, and the SCAs. The one thing that it's important to notice is for the genomic models, we have the genetic or genomic relationship matrix within each effect here for females, for males, and for the hybrids. For non-genomic models, instead of having this genomic relationship, we have just an identity matrix to make sure that we have our data structured. And the G/E was calculated by the Hadamard product between all the genetic effects and the environment. And all the analysis were done using Bayesian generalized linear regression package in R. To validate our models, we used a leave one out cross-validation scheme where we predict hybrids where we didn't have that female. And this we're going to call it a T1. And predicting hybrids with unobserved males. So every time that male were removed, hybrids where that male or that female was included, we would remove, and then we'll try to predict those. And then at the end, we're going to remove both. We got a smaller portion of the data, and we did the same cross-validation scheme, but we used the Texas hybrids, and we predict the performance of Texas hybrids using data from Texas, and then we did the same thing using data from Kansas. And we did the same thing for Kansas hybrids to see if the adaptability would help increasing the performance of the models. And we're going to use the Pearson's correlation to calculate the prediction accuracy of the model. So we can see some of the results here now. And for grain yield, here are all the blues that we used in order to create our genomic prediction models. Plant height, the same thing. This is in kilograms per hectare. This is in centimeters. And days to one thesis. We see that the COVID-19 was the best environment, and we see some variation here. So G/E probably is extremely pronounced, so we need to account for that. And on the other side here, you can see the repeatability and the percent of coefficients of variation, saying that the quality of our experiment was pretty good. We have relatively high repeatabilities for grain yield, for plant height, and also days to flower. And the red color tells you the percent of coefficients of variation, which tells you the quality of the data for that specific environment. So from the combined analysis, we can see that all the genetic effects and the G/E are extremely significant. And we also see that most of the variation is explained by the environment. For the combined analysis, we also calculated the repeatability, and we see that the data was pretty repeatable. So it is a good data set for us to play with a little bit. With the genetic distances based on molecular markers, we see that there is a clear cut between the heterotic groups of females and males. And another thing that is easy to see here, before I should say that we were able to explain almost 45% of the variation using the first and the second principal coordinate. And we can see here that the variation within the male is much more evident than the female because of the time that we have to apply to develop A lines. We have a process to sterilize new A lines, and that takes more time, and the variation within this group is a little smaller than the males. Now we're going to see the prediction accuracy for non-genomic and genomic models for a single environment, multi-environment, and then for a G/E model. So BRR stands from non-genomic. And we can see that we are actually able to predict the performance of hybrids without using genomic information. We see that females and males, so here we see the performance on average on the females, and we see the performance on average of the males. And this is to predict hybrid again. This is just using a different cross-validation scheme. And I want you guys to look here at the blue values, and you can see that the best model was for the genomic model using the G/E. This is for grain yield. One thing that is interesting to know to notice is the prediction for the genomic model. It's interesting to see a pattern here that it's easier to predict hybrids when we have the cross-validation involving females. And again, because they are close related, we can extract more information between that gene pool, between the females. So if you compare within the model, the A lines are always producing higher prediction accuracies to predict hybrids. The same pattern is you can see in plant height and also day-1 And this single environment was just for college station. And we asked, okay, so let's see the prediction accuracy within each location. And with this picture here, it's important to see that, for example, here in Hayes, we see a big drop on the prediction for days to flower, and that's because we only collected data for days to flower in that environment on the first rep. So repeatability is extremely important for you to predict, for you to develop your prediction models. One thing that is interesting to see is so females have a higher prediction capacity to predict hybrids in the South, where the Texas environments are, and it's interesting to see that males have a higher prediction accuracy or ability to predict hybrids in Kansas environments. Now we're trying to see if the envirotype phenotype covariance actually explain what happened in this cross-validation scheme. So again, we have the Texas by Texas hybrids in Texas. So when we see that when we use data from Texas to predict hybrids, to predict Texas hybrids, we have higher prediction accuracy than when we use data from temperate environments, which is Kansas environments. And the same trend is also observed when we have Kansas hybrids, data from Kansas and predicted in Kansas. So this is interesting. And this is likely due to a covariance that exists within those lines that were adapted to those locations. And the literature talks about it, and they call it a variotype-phenotype covariance that is innate to the genotypes that are developed within a target environment. So how can you deploy a genomic prediction into a hybrid program? Usually you can test all the possible hybrid combinations, right? So you would have a subset of lines, you would cross them, and then you would predict all the untested hybrids. And then you would select only the ones that are expressing higher higher yield potential, and then you would test those hybrids in later stages. And that was proposed by Techno in 2014. So how we would do that in hybrids, in sorghum, sorry. We would, to develop the genomic prediction model, we first would replace the test crosses with a factorial design. And we would make this crosses, and we would use as much R lines and B lines we're able to. Then we would use a factorial design and develop these genomic prediction models. And then once the model is developed, we would start applying genomic selection to find our advanced lines, to predict the lines, and put them back into the breeding crosses for each heterotic group. And also we would be able to screen for hybrids that would express the potential for later development. And these B lines would then pass through sterilization, but that's the idea. We would screen hybrids, and for us to keep using the model, we would use the data from those advanced hybrids to update our model. And I don't think that in this scheme here, testers will have the same importance as they have it today. So to conclude, genomic and non-genomic models can predict sorghum hybrids. Genomic models provide more accurate predictions. More complex model, including G/E and population structure, should be considered. Several factors affect genomic prediction models, and genomic selection should be implemented in grain sorghum breeding programs. We have some future projects that are being under evaluation now, and it's just getting the kind of the data set I had, but increasing the numbers. So now they have an unbalanced data, and they will validate some of the models that I presented here today. And also we'll be including some other sources of variation like environmental covariables to try to increase the prediction accuracy of our models. And to finish, I would like to acknowledge all my committee members, Dr. Rooney, Dr. Patricia Klein, Dr. Ramasamy, Dr. Dabney, all the guys here from Sorghum Group, Delroy, Vicky, Dr. Hoffman Jr., Dr. Carvalho, Dr. Gomez, Dr. Pfeiffer, Dr. Pugh, Dr. Horn, Dr. Harvey, and future doctors, Amani, Daniel, Mitchell, Fabian, Noah, Kayla, and Nick. I would like to extend my acknowledgements to the molecular lab Dr. Robert Klein, Julie, that helped me a lot with extracting DNA, Dr. Paddle, and Lauren. And all of the student workers, because they are extremely important in this program. And I would like to acknowledge all of these sources of fundings that somehow helped me to finish this, not finish yet, but to be here. And Dudley Smith Family Travel Scholarship gave me the funds to go to Mexico and actually learn some of the genomic prediction models, how to develop them. And with that, I would like to take any questions. This is my email. If you want to contact me for later talk, I'll be happy to. Thank you.
Great job. Great job. Very interesting stuff. I'm sure there's lots of questions. Go ahead.
I actually have a question. So with, I guess, everything you know about modeling, how do you see like sorghum breeding programs progressing in the future, say like 10 years from now? Do you think everybody's going to take this and implement it into their programs, or what are your thoughts?
I think there is a big opportunity for them to start using genomic prediction models because, again, it increases the number of hybrids that you can screen. And if you start doing that, you can increase the selection intensity, which affects directly the rates of genetic gain. And also, once you have developed the models, you don't need to have the early hybrids phase to screen for those hybrids. So you shorten one cycle for selection. So I really believe that they will be using genomic selection to perform selection. And also with the genomic prediction models that people are developing nowadays, they are including environmental covariables that are trying to predict future environments that we, because of climate change that people talk about, they are trying to actually see the behavior of some of those genes or the markers that we have in some particular scenarios to see what will be the best hybrid in a particular environment. So that's something that people are looking for. So I believe that they are using genomic selection. They will be using.
Thank you so much.
You got a question in the chat.
Let me see. Where is the chat? Oh, here. Oh, here. Okay. What was the population size in those? So the population size for the training set.
The total population size. I probably just missed it. I mean, if you got a slide on it, if you could just pull it up.
No, the population size for the training set, we use the whole data set for it. But for our validation, we would chop all the females. So for example,
that's fine. What's the total number?
That would be 10.
How many genotypes?
So we would exclude 10% and we would use 90% of the data.
Okay, so what is the actual number of genotypes though? The N equals.
So the total is 100. 100.
Okay. Thanks. That was my question. Yeah, I think so that's going to be a statistical, a little bit of a statistical toughie on that one. So okay, I'm much clearer now.
You're right.
Okay.
But so it is a total of 100 hybrids, but it was replicated. So in total, it was 10 different environments. So you're looking at 1,000 hybrids, but different hybrids 100.
Yeah, the genotype matters far more than the number of replications. I mean, the replication of the allele is much more important, but that explains some of the results I was seeing. Now I understand. Thank you.
Yeah, my pleasure.
There's another question in the chat from Ziqian.
How much variance is estimated to be additive? So that's a great question. Unfortunately, we were not actually modeling the additive effect, the dominance effect, for the genomic prediction. But if you go back on the phenotypic analysis, is that the phenotypic analysis or the genomic analysis? So no, sorry, let me go back here. I'm sorry. He wants to see this. So probably this is what he wants to see. So most of the variation is due to additive variation from the female and from the male. You can see, am I sharing the screen?
Yeah, we can see it.
So yeah, so here you can see that the SEA is only 1.3% of the total variation, and the GCAs of females and males is this much. And Do you see the difference hybrid population have more specific interaction between the two parents? Well, so the population structure that we were talking about was within that the hybrids that we have. So we were able to separate the females from the males. I think that's what he was trying to ask. Does hybrid performance help to shape? Oh yeah. So the hybrid cross would allow you to estimate the general combining ability of the male and of the female, which is the value of that line in hybrid combination. And historically, that's how sorghum breeding programs is being improved. The yield has been increased based on GCA. So yeah, that definitely helps us to improve the performance of hybrids and the performance of lines of the whole program.
Other questions? Going once, going twice. All right, thank you very much to our speakers today for 2 excellent seminars. And we'll see you back here, same time, same channel next week on the Texas Soil and Crop Sciences Hour of Power. Ciao.